Hi, everyone, and welcome back to Immunology Unplugged, the podcast where we decode the immune system and talk about what's really going on in your body's microscopic defense force. If you haven't listened to our first episode, I highly recommend going back and checking it out. We kicked things off by diving deep into the T cells, the immune system's elite fighters, and we explored how they're involved in everything from cancer therapy to autoimmune diseases and even brain health. And because today we are going one layer deeper, we're not just talking about what T cells do, we are also talking about how we watch them do it. So, welcome to the world of flow cytometry, the colorful high speed technique that let us watch the T cells in action and in real time. So, for those who don't know what the flow cytometry is, imagine this: you have millions of cells in a tiny tube. And some of these cells are T cells and some aren't. Some they are activated, some are exhausted, some are making cytokines, and others are just floating by. With flow cytometry, we can take that mix and analyze each cell individually, one by one. As they fly past a laser beam at high speed, each cell is tagged with fluorescent markers, glowing labels that attach to specific proteins like CD3, CD4, CD8, CD45RA, CCR7, and more. So when the laser hits the cell, it emits a light signal, and that signal gets turned into data points, graphs, and plots. It's a very fast technique, it's powerful, and it's the gold standard for tracking immune responses. So the question that will be asked here is, how do we identify T cell subsets? Flow cytometry is a kind of like immune system detective work. So you start by tagging the T cells with surface markers, as I mentioned before. CD3 is going to tell you that, yes, this is a T cell. CD4 versus CD8 will split your T cells into helper and cytotoxic groups. Of course, the CD4 is the helper, CD8 is the cytotoxic, just from the name. The CD4, T5, RA, and CCR7, they will tell you if they are naive, central memory, or effector memory cells. Then we have also other signs that we can detect our T cells, like the PD1, TIM3, LAG3, that are the signs of exhaustion of these cells. And then we have when these cells are activated. How can we know that? It's specifically by using CD69 and CD25. So you can even go inside the cell using intracellular staining to detect the cytokines like interferon gamma, IL-2, TNF-alpha after stimulation, depending on what experiment you're doing and depending on the question you want to answer, of course. That means that we don't just know who the T cells are, we also know what they are doing, which is really cool. So how to read the flow cytometry graphs and not to be scared of them. I know a lot of beginners who have always this feeling of like being really anxious and not really understanding what's going on in these graphs. I promise you I will make a video talking about that and explaining to you every inch of these graphs because I've been there, and I hope it will help many people and many students in research and also PhDs and masters and bachelors. But here's the secret that I've learned so far from my very humble experience. Each dot you see on a flow cytometry plot means a cell. Each small dot is a cell. So these dot plots, they show two markers at once. For example, the CD4 on the x-axis and CD8 on the y-axis. You'll often see four quadrants: CD4 positive, CD8 negative, which are the helper T cells, and CD8 positive, CD4 negative, which are the cytotoxic T cells. So double positive, double negative. The histograms show how strongly one marker is expressed. The further right the curve, the higher is the expression. You try to imagine with me. I will try to put on a figure if I have like the exact figure that explain exactly that. So the gating strategies help you filter the data. First, you select live cells, then the lymphocytes, then CD3 positive T cells, and so on. This is actually projected on every person who's actually investigating the T cells and using the facts. Don't worry, I will be posting short reels explaining the facts. Then we have like real-world applications. Flow cytometry isn't just a lab technique, it's used in clinical trials to monitor immune responses to vaccines and therapies, in autoimmune research to understand dysregulated T cell subsets. It's used also in cancer immunotherapy, especially in the CAR T cells, where we are tracking their activation, their exhaustion. It's also used in COVID-19 research to study the T cell responses during infection and recovery. So it helps us watch the immune system response in real time, and that's incredibly powerful for both research and also medicine. So basically, flow cytometry gives you a window into the immune system that no other method can, which is amazing. And we're not just guessing what cells are there, we're seeing who they are, what they're doing, and how they're feeling, all in a single experiment. Good. So that's it for today's episode of Immunology Unplugged. If you found this episode helpful, please share it with somebody who's learning about immunology or flow cytometry or T cells, or someone who's just started a dot plot and had no idea what they are looking at. Don't forget to check out episode 1 if you haven't already. It's a perfect primer on T cells and why they are such a big deal in science and medicine. And keep an eye out for the upcoming videos on how to read flow graphs. I will walk you through it step by step, no jargon, just clear insights. Thank you very much for listening. Until the next time, stay curious, stay critical, and stay plugged in.